A new molecular pathway that controls lifespan and health in worms and mammals has been identified by researchers. The researchers showed that microscopic worms with excess levels of these proteins live longer and healthier than normal worms. They also show that mice with excess levels of these proteins demonstrated a delay in blood vessel dysfunction associated with aging. This study has major implications for the understanding of aging and age-associated disorders such as heart disease and dementia. This study, conducted by researchers at Case Western Reserve University School of Medicine and University Hospital's health system, involved experiments where the proteins in question were artificially increased or decreased. We find that by artificially increasing or decreasing the levels of a family of proteins called crumple-like transcription factors, we can actually get these small worms to live for longer or shorter time periods, said first author Nelson Shea, MD PhD fellow at Case Western Reserve University School of Medicine. Since this same family of proteins also exists in mammals, what is really exciting is that our data suggests the KLFs also have similar effects on aging in mammals too. The researchers found that KLF levels decrease with age and that sustained levels of KLFs can prevent the age-associated loss of blood vessel function. This is intriguing given that vascular dysfunction contributes significantly to diverse age-associated conditions such as hypertension, heart disease, and dementia, said senior author of the study Mukesh K. Jain, MD, Professor, Vice Dean for Medical Sciences at Case Western Reserve University School of Medicine and Chief Scientific Officer of University Hospital's health system. When the researchers conducted further analyses, they found that KLF proteins work by controlling autophagy, a recycling process that cells use to clear debris such as misfolded proteins or normal molecular byproducts that build up in old age. According to the researchers, the loss of this quality control mechanism is a hallmark sign of aging. As cells age, their ability to perform these functions declines, the authors wrote in their study. This likely leads to an unsustainable accumulation of toxic protein aggregates, which ultimately present an obstacle to cellular survival. Indeed, worms without KLF proteins cannot maintain the autophagy process and die early. According to the researchers, the next step of the research will be to study the precise mechanisms underlying how autophagy in cells lining blood vessel contributes to improved blood vessel function. As our population ages, we need to understand what happens to our heart and arteries as we rely on them to function perfectly later and later on in our lives, said Shea. Our findings illuminate what can happen during aging and provide a foundation to designing interventions which slow these processes.